don't need them in particular. Okay, I need to share my screen, put up my chat. So go ahead and do the seating chart. The seating chart is up and ready to go. I did open the homework earlier today. Somebody sent me an email, so it should be open. Is it not open? Go and check right now. Only one person sent me an email that chapter 10 homework wasn't open yet, but it's open now. It should be open now. Put it that way. Okay. Magically, it's open. Poof. Perfect. Awesome. All right. No, we did that. Nice. It's so hard to see. Control points. Here we go. All right. Okay, how are we doing? So, show of hands, how many of y'all attempted these questions? Good. I'm very, very proud of you all. Very proud of you for attempting them. Some of them we're going to discuss the material today. So it was probably good for you to go and read about it, right? So that we can discuss it. So what we're going to do is we're going to finish the chapter and then we'll have the rest of the class. And it's really just a couple of slides about regulation. And then we'll talk about the discussion questions and their answers. I think I started it and I think I didn't log into it. Let me see. Oh, I didn't. Thank you. So many things. Nichols.com. And our joke. Yes. Oh, maybe it didn't like my capital B. Okay. What? Oh. No wonder. Okay, there we go. All right. So what did the dog say when he sat on sandpaper? Oh, that's rough. I laughed too hard at that one. Okay. So iClicker is open. Y'all are welcome to get on there and please take care of that. Remember, if you're having an issue with iClicker, swipe up, completely close iClicker, get out of it, and then go back in. I always recommend you swipe up at the end of class to make sure that it's not running in the background because then it causes issues. Okay. All right, so now. So a couple of different announcements before we really start. If you're in my lab section, I have graded lab reports sitting on the outside of my door for you to pick up at your leisure. That some people picked up before break, but a lot of people didn't. Because that was the lipid lab to pick up. I'm still grading the protein lab. Chapter 10 homework is open. It is due Thursday night. It is due Thursday night because we have what on Friday? An exam. Exam 4 is on Friday. What is exam 4 over? Just 9 and 10. It's just glycolysis and citrate. Isn't that kind of awesome? No, like no. All right, and then we sent out, well, I sent out to my lab section. I don't know if Dr. Smith sent an email out to y'all yet, but the lab final will be Tuesday evening, December 7th. We're going to open it for a 4-hour window from 6 p.m. to 10 p.m. and you'll have 2 hours to take it in. And it'll be on Moodle. Okay. So everybody will take it at the same time, but you have a 4-hour window open. And then you won't have that during like regular exam week because we're going to end like a week early, basically. Cool. Questions? So you don't know the exact time? No, it's going to be open from 6 p.m. It's okay, from 6 p.m. to 10 p.m. So that's okay. So it's a big 4-hour window, but you only have 2 hours to complete it. So if you start at 6, it closes for you at 8. If you start at 7, it closes for you at 9. Right? Make sense? Okay, good. Yes. No, it will not be all multiple choice. It will not. You should absolutely have a calculator. You should absolutely have your lab notebook. You should have some paper that you can do some math on. You know, are you talking lab exam or my exam 4? No, oh no, it won't be all multiple choice. It was nice though, wasn't it? You know, but good try, good try. All right, and the exam the lab final exam is also not all multiple choice. There'll be discussion, there'll be problems, things like that, and there'll be multiple choice. Anything else? Yes. I'm sorry, what? What is the lecture final exam? That's a really great question. So you can go on Moodle. What are y'all saying? No. I have it as the 14th at 1 p.m. Yeah. Is that right? Let's make sure it's right on the calendar.
That's what it says on the calendar.
That's what it says on the calendar. See, y'all's internet's faster than mine. Okay. If I could go to the right place, here, class calendar. So, right, all the way, here we go. So Tuesday from 1 to 3 p.m. So that's Tuesday, that's 13, 14, Tuesday the 14th. Okay, yes?
Can you give us any information like how you're going to structure the final?
Similar to this. So the question is, what's the structure of the final exam going to be? It's going to be similar to a regular exam where you have multiple choice, you have fill in the blank, and you have discussion and some problems. So it'll be very similar to what you've had in the past, but it will be cumulative.
Is it going to be the same length as a normal exam?
It'll be a little bit longer. Be a little bit longer. Anybody else?
Have you talked about like the percentage of each thing?
No.
Will it be like more of chapter 11?
There will be, so that's a good question. There will be more of chapter 11 on the final exam than any other chapter, just because you will not have been tested on it.
Right.
Otherwise, it'll be pretty equal 1 through 10.
Okay.
So a little heavier on 11, but equal 1 through 10 the rest.
Gotcha.
Yeah. Good. Anybody else? I hope everybody had a good Thanksgiving. And if you're a chem major, I left a great big Tupperware container of jambalaya in the fridge upstairs. So, okay. Moving on. Here we go. Regulation. So when we regulate the citrate cycle, right? Everybody watched the video that you were supposed to watch. It was only like 25 minutes. Wasn't that kind of nice? All right, so when we talk about regulation of the citrate cycle, there are 3 main regulatory enzymes. But before we talk about the regulatory enzymes, I want to talk about how is citrate in general regulated? How is any cycle in general regulated? Like how are the main ways that you can regulate a biosynthetic or any kind of biochemical pathway? Substrate availability. Tell me, for this cycle in particular, what are the 2 substrates that we're worried about? It's a cycle, right? So start me at the beginning of the cycle. What goes in to reaction 1? Acetyl Coa. And what's the last product? Good. Right, those are our 2 substrates that we're worried about their availability. I lost my chat. Here we go. Okay. All right. What is another way that we can negatively regulate a given cycle. We make the products inhibit the initial steps. What do we call that? Feedback inhibition. So what's our first step? You know, like if you're thinking about feedback inhibition, you can think about the very end of a pathway, or you can just think about the very first reaction. So that very first reaction, what enzyme is in that very first reaction? On the screen, hint, hint. We call it the citrate cycle because the very first product is citrate. So what enzyme makes citrate? Citrate synthase. Do you need to study your pathway? Yes, you do. Okay. So citrate, so here's our example. I'm just going to give you an example, right? What is going to inhibit citrate synthase? What do you think? If citrate synthase, this makes citrate, C-I-T-R-A-T, citrate from Acetyl Coa and oxaloacetate. So what should inhibit citrate synthase? Citrate. So citrate, C-I-T, oh my goodness, C-I-T-R-A, citrate synthase, oops, citrate inhibits citrate synthase. Right, that's an example of feedback inhibition. Do we want to have very early feedback inhibition? Absolutely we do, right? We want to stop the citrate cycle from happening if we've already overloaded the cycle, right? If we're piling up citrate, then that means that the whole cycle's backed up. Maybe we've made a ton of ATP through oxidative phosphorylation. We don't want to do it again. That's a waste of energy. You need to go and prepare for winter, right? And go shut off that acetyl CoA to make fatty acids, right? So you got to think about these pathways in terms of not only themselves, but other pathways that it's connected to. And it will help you to understand the pathway better, okay? All right. And then we have feedback inhibition, and then we have another form of feedback. We have feedback control of key enzymes, right? So what are the 3 key enzymes that are regulated in the citrate cycle? Citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. So what do we mean when we say feedback control of these key enzymes? Well, what things are going to regulate all 3 of these? What do we want to be able to regulate all 3? So all 3, so this is really this, right? All 3 regulated by what? What is the very end of our pathway? We go to the citrate cycle, we go through the citrate cycle, we make NADH, FADH2. Where does that NADH and FADH2 go? Electron transport chain, oxidative phosphorylation, and we generate what? ATP. So think about the 2 end products of those 2 things, right? The end product of citrate cycle in this context is NADH and FADH2, right? And the end product of oxidative phosphorylation is ATP. So we want the end products of these 2 things to come back and inhibit our cycle, right? Because we're saying basically we have a high energy charge in the cell. We don't need to make any more. We're full. So they're all regulated by NADH and ATP. that make sense? We want the end products to inhibit different parts of the cycle. Okay. Yes. No, because they're not all regulated by FADH2. They are all regulated by NADH and all by ATP, if that makes sense. Okay. All right. So if we kind of look at our cycle, right? Normally we have pyruvate. Here's pyruvate. Pyruvate undergoes our pyruvate dehydrogenation. So it goes through that big complex, right? And we make acetyl Coa. Now, if we have acetyl Coa and we have oxaloacetate, we can go through our very first reaction and we can make citrate, right? So look at those first two reactions. So when we go from pyruvate to acetyl Coa, pyruvate dehydrogenase is also regulated. This is not a part of the citrate cycle, right? We all know that. It's separate from the citrate cycle and it is a separate step from glycolysis. So if you look at that pyruvate dehydrogenase, now look at what's inhibiting and look at what is driving the reaction. So we said NADH and ATP are all inhibitors within the citrate cycle of the three key reactions: citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase, right? Those were the three we talked about on the previous slide. But look at what we have in common with that conversion of pyruvate to acetyl Coa. What can inhibit pyruvate dehydrogenase? NADH and ATP, and also acetyl Coa. But look here, NADH, ATP, NADH, ATP, NADH, ATP, right? Do you see that these are the two that are in common for everybody? And that really makes sense because this is what is driving the reaction to happen, right? It's that energy charge of the cell, whether you have ATP or ADP and AMP, right? Or if you have NAD plus versus NADH. So it's important, I think, when you're looking at these big pathways, is to find commonalities so that you understand that regulation, because that's where the commonalities come in. Okay, so now you can look at some of the differences too, right? So there's another regulatory molecule, right? If we are particularly in muscle cells, how is regulation going to be different of the citrate cycle? What do we need in order for muscle cells to contract? We need calcium. So look at what is a positive regulator of most of those reactions, not all of them, but most of them. Calcium, right? And we need that to drive muscle contractions. And we skip the muscle contraction part of whatever chapter that was. So you don't know a whole lot about it, but you do know that calcium is required. Okay, so when we look at this reaction, right, we also need to think about what happens if you're getting acetyl Coa from a different source other than glycolysis. Where can we get acetyl Coa if we don't get it from glycolysis? Where? Fatty acid oxidation. Fatty acid oxidation will make acetyl Coa as a product. So let's think about this, right? If we don't eat a whole lot of carbs, right? Then our body is going to switch to metabolizing the fatty acids that our body has stored into acetyl Coa, right? And that's great. But if you think about this, really we need two things to drive the citrate cycle. We need acetyl Coa and what else? What? oxaloacetate. So how in the world can we get enough oxaloacetate in order to drive the citrate cycle when we have a surplus of acetyl CoA? We can use a separate enzyme, and the enzyme that we use is Pyruvate Carboxylase. We can convert pyruvate straight to oxaloacetate. So in fatty acid oxidation, you have high, very high acetyl CoA. But you can't keep up. You don't have enough, you have not enough oxaloacetate. ACETAT, oxaloacetate, right? So in response, you use pyruvate carboxylase in order to convert pyruvate to oxaloacetate. Now, do we have enough of both of the two substrates? Absolutely. Absolutely. But this is only going to happen when we've made enough ATP, right? You don't want this to happen if you don't have a whole lot of ATP around. Okay. All right. When you look at this cycle, we can think about using this cycle in terms of energy production, which is primarily how we've been discussing it so far. But that's really not the only use. There are actually, industry-wise, some other ways that we can basically hijack this system. So if we look, y'all know one of the first product, right, is citrate. But what's another name for citrate? Citric acid. How often do we use citric acid in our daily lives? It's massive in food preservation, right? I mean, you see that on food labels for half of the different things that you eat. It's actually used in polymer chemistry. So a lot of times you need a reactive acid in polymer chemistry. In pharmaceutical medicines, it's used quite a bit. And so what we actually do is we use a mold. We use an aspergillus mold in order to produce a ton of citrate. And we do that because we know that this organism is going to go through the citrate cycle, but we're going to select for specific organisms that do not use the citrate in metabolism, that instead of using the citrate in metabolism, they are actually going to export the citrate. So citrate is made where? Where are we? What organelle are we in? We're in the mitochondria, right? So we actually have to make two different exports in order to use citrate industrially in a high enough quantity. So you're actually going to select for isolates of this mold that will export citrate from the mitochondria and then also export it into the culture media, right? And then all you have to do is harvest that culture media and it is super rich in citrate. And the very last thing that you do is you make sure that that citrate does not get metabolized back into either acetyl CoA or oxaloacetate. So you inhibit the citrate lyase that would catalyze that reaction. And then we can make tons of citrate, tons of citric acid. And it's really, really important for industry. So there are multiple reasons why it's super, super important. Okay. All right, so when we talk about citrate, we really need to talk about, gosh, how connected it is, right? So we know that the citrate cycle makes our redox power, right? So citrate makes what two energy carrying molecules that are used in redox? I'm not talking about GTP here. What are the two molecules? NADH and FADH2, right? So we know that citrate makes those, and those can go on to the electron transport chain and participate in more redox reactions. That's true. But that's not the only thing that can happen. So when we look at the citrate cycle, it's really important to see that there are other substrates within the citrate cycle that can be used in other pathways. So we really call the citrate cycle an amphibolic pathway because it can be catabolic. What we've described so far has been primarily catabolic, right? We break things down. But we can also build things with intermediates from the citrate cycle, which is why we also call it anabolic. So let's look at these different things. So the catabolic pathway, this is where we oxidize, right? Acetyl Coa, acetyl Coa. And we're going to make NADH, FADH2, right? That's the whole cycle that we've kind of been talking about so far. So we already know that pathway. But when we talk about anabolic, we're going to talk about a bunch of different things that citrate can make. Citrate can make amino acids. We can go through fatty acid biosynthesis, and we can go through heme biosynthesis. So there are two places within the citrate cycle that we can have intermediates that will go and form amino acids. So if we look at that, we're looking here at oxaloacetate, and we're looking at alpha ketoglutarate. Either one of these two intermediates can go into an amino acid biosynthesis pathway. So those are two ways that we can shunt off some of these substrates to go and do other things, right? So once our energy charge gets high enough, we have to make amino acids. We have to make heme. We have all these other things to make. And so we will go through and do that. Heme biosynthesis is from succinyl CoA. So what do we need heme for? What is heme? What do we need it in? Hemoglobin in red blood cells, right? Super, super, super important. Now, how do we make fatty acids? Well, where do our fatty acids come from? Acetyl CoA, right? Because if we oxidize fatty acids, what do we make? Acetyl CoA. So we use acetyl CoA in the synthesis of fatty acids. So that's the other one that we need to talk about. And you can also use acetyl CoA to go in and make cholesterol, right? And it's kind of important to remember you can go back up the pathway of glycolysis, right? It's not glycolysis anymore. If we go the back way, what do we call it now if we go in reverse? Gluconeogenesis, right? So you can do that from oxaloacetate as well. So you could actually make sugar as well, going from oxaloacetate through gluconeogenesis. So there are lots and lots and lots of different things. So glycolysis was a very straightforward pathway. Citrate is a little more intricate and a little bit more connected to different pathways, right? Okay, so let's look at some of the reactions that are going to actually replenish the citrate cycle intermediates that get shunted off, right? Because if they get shunted off, you're going to have holes in this cycle. And when you have holes in the cycle will stop. So then how do we replenish the holes? Well, we have two major pathways that are Anaphoratic reactions. So these are any reaction that replenishes a citrate cycle intermediate, right? Because we've shunted some of them off to go make amino acids. We've shunted them off to go make heme, whatever it is. Well, we can use Pyruvate Carboxylase, which we talked about just a minute ago, to convert pyruvate to oxaloacetate. So where was that in the cycle? Right, ooh, went too far. Pyruvate carboxylase, right? We can convert pyruvate to oxaloacetate. So let's say that you pull out alpha ketoglutarate to go and make what? What is alpha ketoglutarate made? amino acids, right? So if we take out alpha ketoglutarate, are we going to go in and make succinyl CoA? Are we going to go in and make oxaloacetate? No. So how do we replenish this pathway? How do we make sure citrate can keep going? We use something like pyruvate carboxylase to create that end product of oxaloacetate. Kind of make sense? Kind of have to think about that for a minute, right? Okay, so let's look at the next one, right? And this is another one that requires other cofactors in order to run, right? So this one actually requires biotin, so that you'll see it like that in the reaction. Another way that you can replenish oxaloacetate is phosphoenol pyruvate carboxylase. What do you notice about both of these two enzymes? They're both carboxylases. They are both carboxylases. And so what we can do is we can take malate. We can convert that to pyruvate, then pyruvate, phosphoenol pyruvate, and then phosphoenol pyruvate carboxylase will take that back to oxaloacetate. So you just have to remember that there are ways to replenish the cycle that are not inherent in the cycle itself, because you're shunting off those intermediates to other pathways. And you need to make sure that citrate can keep going. Okay. All right. So take out your questions. I need to pull that up on mine. So these are the ones you were supposed to work on in place of last Wednesday. All right. Are y'all ready to answer them, or y'all want a few minutes? What do you think? Do you want a few minutes to talk to your neighbors, talk it out, or you want to talk it out as a class? Neighbors first? Okay, so it's 11:16 because we have a lot of time. So I'll give y'all, how about I give y'all, that would be 26, 36. So how about I give you 10 minutes or 8 minutes? Let's do 8 minutes. We'll do 8 minutes for y'all to talk to your neighbors, compare what you have, and then we'll come back together. So 8 minutes. So it's 11:17, right? 3, 5, so 25. We'll come back together at 25. So, but I do expect y'all to turn and talk to people. And if you need the questions, they're up here. Anybody need questions? Didn't print them out. Anybody else? Some type of feedback. It's always some type of feedback. You know?
No, I just okay. I'm sorry.
Absolutely. So go back and watch the recording for the end. Yes. Yes. Tell her. I hope it's a quick recovery for her. Okay, and then I'm going to pull up. I don't want to pull up because that's got to be it. Maybe. Okay, so this is what everybody's working on right now. And then there's a part 2 questions as well.
Oh, yeah.
Acetyl Coa itself has two. But if you talk about it turn it into acetyl Coa, and then what can it go and do? It actually has way more than just space. So, oh wait, I really want to do like this stuff. So if you think about the two phase that just I didn't think one thing was like, oh my god, what was, and then pick it out, big everything. I have acetyl Coa, the only thing, and then deconverted. What are all the things that can happen? There were just a few parts that I'm like in the cell, right? Like it can get turned into something, which then can go this way or go this way, you know, not just acetyl Coa. So it kind of depends on how you're thinking about it. We'll talk about that in a second. I don't know. I'm gonna put iron. It's very fun. And then I had like 6. That's the one. I'm trying to get the lab report done. So if I don't have it done, the enzyme has to make the hydrogenase. So that's different one we have in the last video. There's like 3 and 4 things. But it doesn't matter. It takes so many PhDs. I mean, you can't tell. That's a whole lot. Because your brain's going too fast. The only part but the only part I got was this draw a really good picture about it. So you drew a really good picture, so that should help me like.
Yeah, one thing y'all can do is like take the cycle that you have and then add in regulation. Add in, okay, what happens if this is shunted off? You know, like take a core picture and then add things to it. Yeah, that's a good thing.
Here we go. It's on. I don't know which line I'm, but I got it. I'm sorry, y'all on Zoom. It's class discussion going on, but we're going to come back at 11:55. 11:25, not 55. Haha.
There's always 7.
What time we got? Okay, one more minute. Has it really been that long? I do have it. I do have it kind of nice. I want to add some things, but I do have it kind of nice. But I'm not posting it. I'm not posting it. Y'all about ready? I guess we're still waiting 1 minute, huh? Let me see the clock now. Come on, phone. All right, y'all ready? Everybody got quiet. Okay, so the first one was about driving redox reactions in the cell, right? And when we talk about redox reactions, what are we talking about? Energy carrying molecules that are carrying what? Electrons. So why do electrons move? That's really explaining what drives a redox reaction, is why electrons move. And when electrons move, it's because of differential reduction potentials. So remember that table that we looked at in class, right? And which molecule had the very highest reduction potential? Oxygen. So have we seen oxygen yet in our pathway? Have we talked about oxygen? No, oxygen is going to be at the very what? At the very last step. So our very last step is in oxidative phosphorylation, right? So we're not going to see oxygen until we get to oxidative phosphorylation, which is going to be the very, very end. But it has the most positive reduction potential. So that's all these redox reactions are, is transferring from one place to another because you have a different reduction potential. So you go from low reduction potential to higher, right? And each time you jump, each time those electrons move, they go to whichever recipient has the higher reduction potential. And you're going to see this a lot when we get to electron transport chain. You're going to see, okay, we have NADH. Now what does NADH do? Well, it's going to drop its electron off at complex 1, right? Then what happens to that electron? Oh, it's going to hop along. Well, why is it going to move? It's going to be the exact same answer. It's a difference in differential reduction potential. Does that make sense? Yes, no, maybe, yeah. Okay, all right. So now, what are the 3 fates of acetyl CoA in the cell? On some papers, you're going to see 2. In some papers, you're going to see 3, because I just couldn't decide what I really wanted to ask. Don't take pictures. Get your pen out. Start writing. So technically, if you're just talking about acetyl CoA, just acetyl CoA, really, you have these 2 major fates, right? If you are simply talking about acetyl CoA, either acetyl CoA can get metabolized into things that will enter the citrate cycle, right? And we're going to drive the citrate cycle. Or we're not going to use it and we're going to shunt it off to fatty acids. But if you expand that picture to what are the 3 fates of acetyl CoA in the whole cell, right? So acetyl CoA turns into what? Citrate, right? Citrate goes in and turns into what? Study your pathway. Isocitrate, right? That first reaction is just an isomerization. Then we go to alpha ketoglutarate. Oh, what does alpha ketoglutarate turn into? What can it be shunted off into? Amino acid synthesis. So we know that it's not specifically acetyl CoA that's turned into amino acids, but one of the products of acetyl CoA metabolism is alpha ketoglutarate. And the alpha ketoglutarate can then turn into an amino acid. What else can turn into an amino acid? Oxaloacetate. Right? So are those the only things that can happen with acetyl CoA? No, right? What else could happen? We could use it in synthesis of what? Sure, cholesterol. What else? Heme. What else? We could convert it to oxaloacetate, and oxaloacetate could also be used to go through gluconeogenesis to make sugar, glucose. Right? This is gluconeogenesis. So there are lots and lots and lots of different fates of the products, right? Specifically acetyl CoA, only 2. But really there are lots and lots of different things that could happen. And that's a really important designation to make. Y'all with me? Yeah? Okay. All right. We did this question kind of in class already. What are the required components of pyruvate dehydrogenase? Remember, it's that complex, and it has 3 subunits. And what are the 3 subunits? E1, E2, E3. But you need, in those 3 subunits, 5 different cofactors. And we call those cofactors vitamins, right? So NAD+, FAD, CoA, thymidine, pyrophosphate. Now the alpha lipoic acid we make. So it's not technically a vitamin, but it's the only one that we can actually make. So what makes it a vitamin? We have to eat it. We can't make it. We have to eat it. So some organisms have to be able to make it. Organisms that we eat on a regular basis have to be able to make it, but we don't. So why don't we make it anymore? It's a waste of energy. Usually they're huge molecules. It's a waste. Why make something that we normally eat? The only problem begins when we have people who don't have access to a healthy diet. And when those people don't have access to healthy diets, what happens? You have a vitamin deficiency and it presents as a disorder. Yeah. Okay. And y'all are supposed to just be checking your answers, right? You're supposed to have all these answers. Everybody's like, yeah, sure. Go slower. All right. So we know pyruvate dehydrogenase is super important, right? Is it in glycolysis? Is it in citrate? No. It is the enzymatic reaction that is between citrate and glycolysis, right? Okay. So why does it go? Because its delta G value is extremely negative, right? Negative 33. So it's going to go whether you have a lot or a little substrate. What's the substrate? We go pyruvate plus what? What are we going to make? We're going to make, so I'll put the products at the bottom. We're going to make acetyl CoA, but we make other things too, don't we? What else do we make? Remember, it's a dehydrogenase, so we're going to lose a what? Well, yeah, that too. That too, yes. So where do we lose the hydrogen to? NAD plus picks it up, and we get NADH plus and H plus, right? Okay, but we're missing something in the reactants and the products. CO2, that's our release of carbon, right? And what else do we need to add here? CoA, right? So we have reactants going to products. So it's not necessarily regulated by the amount of pyruvate there. If you have pyruvate and you have CoA, you're going. It doesn't matter how much you have of it. That's a really high negative delta G value. So how is it regulated? Through allosteric regulation, right? This is what we talked about today. NADH is a negative regulator, right? What is that telling us? If we have lots of NADH, what is that telling us? Well, we're going through citrates so fast that oxidative phosphorylation can't keep up, right? So if oxidative phosphorylation and the electron transport chain is not doing its job, well, we don't want to keep going through citrate and making more and more and more, right? So we kind of want to slow the citrate cycle down, put the brakes on the citrate cycle. So that's a really good thing. Acetyl Coa, right? Acetyl Coa is what? An immediate feedback inhibitor, right? It's still allosteric, but it's an immediate feedback inhibitor. Just like what we saw with what enzyme today that we talked about. Citrate synthase, right? Citrate inhibited citrate synthase, right? That was the immediate product inhibiting the enzyme. Been a couple of days since we talked about this, right? Okay, ATP, we said, right? ATP is kind of one of those universal regulators for basically everything in metabolism, because it tells you you have lots of energy in the cell already. So you need to go and put that stuff into storage. The cell's high energy enough. Go make something else. Go make amino acids. Go make fatty acids. Go do something else with that energy. And then our activators are basically the opposite, right? So those are allosteric regulators, but that's not the only way that we can regulate pyruvate dehydrogenase. How else can we regulate it? Covalent modifications, remember? So when it's phosphorylated, it's inhibited. When it's dephosphorylated, it's active. Okay. All right. Y'all need a second? What do we have? Not too much left. Y'all ready for me to switch or not yet? No, we're saying no. But try to answer these questions on your own. That's why I sent them to you early. Because if you try them on your own, it's going to help you so much more than just copying down my answer. It so will. All right. And if at the end of class y'all want me to go back when everybody else leaves, I could do that for you. All right, so which reactions catalyze oxidative decarboxylation of pyruvate and acetyl CoA to generate 3 CO2s and 3 NADHs, right? So if you hear the words oxidative decarboxylation, you should be thinking I am releasing carbon dioxide. One of them is not in the citrate cycle, right? We said before that is our pyruvate dehydrogenase reaction. Okay, then we have 2 within the citrate cycle. So it's our 3rd reaction of citrate, our isocitrate dehydrogenase. Look at this, they're all what kind of enzymes? Dehydrogenases, yes. So our 3rd reaction with isocitrate dehydrogenase and our 4th with alpha ketoglutarate dehydrogenase. So do you have to know where there are important things going on in the cycle? Absolutely. Absolutely. Okay. How about energetically favorable? And why are energetically favorable reactions important? Because they have very negative delta G values, which makes them irreversible. So what are our irreversible ones? Oh my goodness, they're really similar to what we've talked about, right? Citrate synthase is one of them, but that's really important because that's our very first one. And then look, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, alpha ketoglutarate dehydrogenase. Do you see how there are similarities? Once you learn a few, you kind of got it. You have to see these links. Once you start seeing these links, it's going to make so much more sense to you, right? So sit down with the reactions and think about them. So what are the control mechanisms for citrate? We talked about this earlier today, substrate availability. What are our two substrates? Y'all told me this earlier today, oxaloacetate and acetyl CoA. What controls oxaloacetate and acetyl CoA levels? Pyruvate dehydrogenase and pyruvate carboxylase. That's what we talked about today. Then we talked about product inhibition, where citrate inhibits citrate synthase. We talked about that. And then we talked about all kinds of feedback control of our key enzymes. And that was like what we talked about. That was today. And then what are the key enzymes that regulate the citrate cycle? Well, these are the four that we just listed above, right? It's the same enzymes. So the ones that are highly controlled are the ones that have large negative delta G values, that are under allosteric control, you know, all those kinds of things. And then the other thing is, can you recognize the different reactions that generate either electron carrying molecules or simply high energy molecules? You should be able to do that. You should be able to know which ones are generating what, okay? All right. If you want to stay and copy anything down, you're welcome to stay. Otherwise, you are free to go. And just tell me what you want the screen to be on. To the which page? The first page? E. Okay, there's E. Trying to move that. What are you doing?
Here on Friday because I'm leaving.
So
I'm sorry.
No.
I'm very sorry. I think. Okay, so I think it starts at 11 a.m. on Friday, and I don't get home until like Saturday at I don't know, like 6 p.m.
it's fine. It's fine. I'm not worried about it. No.
But I can take it the very next day I get home, like Sunday.
You let me know.
Okay.
When it works for you, and that's perfectly fine.
Okay.
Absolutely.
Okay, but I'm not letting it have like a week or anything. Like I'm taking it right when I land.
You tell me. Whatever works for your schedule.
Okay.
I will set up on
And then I will be 7 hours ahead of time, but we can calculate it.
Yeah. You tell me. You just tell me what works for you. Okay. And we will make it happen.
For example, when you said the lab final go at 6 p.m., I was like,
oh, wait a minute. I know. Just tell me. You tell me.
Okay.
What works for you.
Okay.
And we will adjust. And look, this is something you have absolutely no control over.
I know, and I called the airline because of course I wanted to change my life, and they were like, "Well, it's gonna cost you $500."
It's the price of a flight.
Yeah.
Yeah, I know. No.
Okay,
no,
I wouldn't need
yeah,
I mean, I'll be here on Wednesday.
Yeah.
But then on Friday,
No
but
problem.
I'll keep it like on.
That's fine.
Okay.
We got it covered. No big
Thank
deal.
you
We
so
will
much.
figure it out. So it will make acetyl CoA because you'll go through fatty acid oxidation first, right? And then once your fat reserves are done, you're gonna start using your protein. So basically where we said you're shunting things off, you can shunt things back in.
So there's three mechanisms to make acetyl CoA: pyruvate, acetyl CoA, and fatty acid acetyl CoA. Would it be also the muscles?
Only under certain conditions, right? Those are like extreme conditions. And so usually we don't say that unless like we're specifying you're under starvation, right? But yes, you can. Yes, you can absolutely make it. No. You have to go through this to get energy. You have to. So in some way, shape, or form, you have to get an intermediate that will enter citrate so that you can go through oxidative phosphorylation. Because to get energy, you need oxidative phosphorylation. You don't have sugar, right? You can't do glycolysis. Yeah, absolutely. Which one? I'm sorry.
H and I.
H and I. That's okay. That's okay. That's why I said, go ahead. I'm happy to do it.
I'm sorry. I had gone through them yesterday, and so I had like all the enzymes. I was just writing down any extra information.
Oh yeah. No,
Because I
absolutely.
didn't feel nearly as in depth. So like,
And that's okay. That's okay. At least you tackled it on your own, which makes a huge difference. Like if you just sit here and get the answer, but if you sit at home and you think about it, and and then just filling in a few missing pieces of information is not bad because you've done it on your own. So that's great. That's great.
Because like I went and I did the questions yesterday, so I went back in the PowerPoint so it was kind of like refreshed on.
Well, and there were some things that we didn't talk about in class until today, so those were kind of difficult for you to be able to answer on the front end. So don't feel bad if you didn't get every little piece, you did great.
And so the exam's Friday, and we have until Wednesday. Are we just gonna like review like?
No, Wednesday we're gonna start oxidative phosphorylation.
Okay.
Because like we have to get to the end of making energy.
So we have like 3 classes covering chapter 11.
Correct.
And then when will the homework be due for chapter 11?
I think I didn't make it due until like right when exams start. So look on the calendar and see. It doesn't matter to me. I could even make it like due the day before our exam. I don't know how I did it. Let me see. No, the answers will not be posted on Moodle. So exam 4 is on Friday because I know on Moodle. Oh, on Moodle it says Wednesday. Hold on, let me see. Let me memorize this. Okay. Okay, so let's go here. Calendar. Okay, so I have it due Thursday, but what I can do is I can put that on. I can put that on Monday for y'all. Then you have like the weekend to do it.
And so the Wednesday's the last day of class.
Wednesday would be the last day of class. Oh, it won't let me change it because I'm not signed in. I will go to my office and I'll change that. I'll change that. But yeah, we'll have 3 days and our exam is on Friday, but it doesn't fit on Moodle. I'm gonna go to my office right now and work on that, making sure everything says the same. Okay. So Tammy, did you get that? Tammy, I don't know if you're still on. Yes. I'm gonna go to my office and I'm gonna go check that stuff right now. But I can't edit it from here because I'm not logged in. So I'm gonna go change it right now. Any other issues?
No, that was it.
Okay. I will see y'all later.